is Ryan Takeshita, research scientist with the National Marine Mammal Foundation, and he's interested in the intersection of cellular and molecular biology, conservation science, toxicology, pathology, and policy. He has provided technical consulting to trustees on several natural resource damage assessment cases, including working on injury assessment and restoration planning for the Deepwater Horizon marine mammal case. At the foundation, he coordinates and conducts conservation science research projects to study marine mammal health and populations, including acting as the research coordinator for the Consortium for Advanced Research on Marine Mammal Health Assessment, also known as CARMA, which is a relatively new GOMRI-funded research consortium.
Thank you. Thank you for the lovely introduction, Chris, and thanks for the invitation to be here at the workshop. It's been a whirlwind 14 hours for me. I've taken a lovely tour of 3 of Dallas's finest airports. So I'm a little loose. This isn't going to be super formal. I don't know if I could pull that off right now. I've been asked to talk with you all today about different types of dolphin health assessments. And first I have to admit that unlike folks like Laura and other folks on our team who have done health assessments for 10, 20 years, we have veterinarians on our team who have spent their whole careers studying dolphins and caring for and saving dolphins. I'm not one of those people. I'm much more like Lori Sullivan, where we kind of got brought in for the Deepwater Horizon natural resource damage assessment, and I've learned fast. And I've learned that it's a whole crew of very impressive people, and so I hope that I can bring that to you all to just kind of share an appreciation for how these folks have created a really impressive, viable science to support decision making. So a little bit about the foundation. Our mission is to improve and protect life for marine mammals, humans, and our shared oceans through science, service, and education. We've been around for about 10 years. We're a nonprofit based out of San Diego. And I think that our mission actually lines up pretty nicely with the goals in the agenda. So we're always happy to come out to events like this. And for a little bit more context about the foundation, one of our main activities is actually working to take care of the Navy dolphin population. And so that means that our veterinarians, our biologists, they have this really unique set of expertise where they've been working with these animals for a long time, and they have this very cool combination of clinical and basic research expertise that they can then apply to helping people make decisions. I'm particularly affiliated with the Conservation Medicine group, and what we like to do is work with all of the top scientists from around the world to turn the basic research that we're doing and the clinical research that we're doing with the Navy dolphins. You can see one here on the left where our lead veterinarian, Cynthia Smith, and executive director is using an ultrasound to assess this dolphin's organs. This dolphin's trained to come to the pier, so it's a really nice situation where in a controlled setting you can develop protocols, you can work out all the kinks, you can make sure that the equipment's going to work well, so that you can then apply this methodology in the field. You can see Cynthia there wearing the super intimidating RoboCop goggles. So this was in Barataria Bay, Louisiana. And so our goal is to help decision makers, especially with situations where we're trying to understand how anthropogenic activities are affecting marine mammals. So like in the Deepwater Horizon ones. Now, by no means did the foundation start health assessments. They've been going on for about probably 20 years now, and this is just a slide showing where, at least in the southeast US, where a lot of those have happened. But what we're really interested in is trying to advance the science that you can do when you're out there working with these animals. So our team has been involved with most of these assessments in one way or another, and especially the ones in red where we were looking at a natural resource damage assessment or an unusual mortality event, and trying to support the research to figure out what happened. And just to reiterate something that I think all of the other speakers have said, we always have to remember that these health assessments need to be done in the context of a suite of research options, right? We can't just do a health assessment. We need to get out there and do remote biopsies or photo ID surveys. Maybe health assessments aren't even the right answer. So when, as a decision maker, when you sit down and you have a question, we need to run through a few different options to figure out, okay, what is the right way to design the study? What are the questions we want to answer? Are you trying to figure out the effects from some kind of toxic chemical exposure? Are you trying to establish a baseline for a future restoration action? Are you, worried about maybe biologists have observed some strange behaviors in a particular population? You want to know more about that. Those are all going to play into answering these first three questions. What animals do you want to catch and study? What do you want to know about them? Where and when can you catch animals even? And so you think through all of these different things with local experts, and then running this reality check by NOAA Protected Resources, right? You want to work very closely with these folks every step of the way. Firstly, to make sure that, is it even worth doing the health assessment? Are there other techniques that we can use that are less stressful on the animals to answer the questions you want to answer? And then second, if you do decide to go the health assessment route, how can you design your study to make sure that you're minimizing the impact that you have on these animals? And of course, the biggest reality check of all, these aren't cheap efforts, and they take a long time to put together. They're logistically pretty difficult. You've got to get 50 to 100 people to a place that's usually in some remote area, and how do you make all of that happen in the timeline that a lot of decision makers have to operate? And so just to reiterate again, every step of what we're going to talk about here, we are thinking about the safety of our personnel and the animals, and trying to minimize the stress that we put on these animals, and always thinking about balancing the responsible sample and data collection with what we're putting these animals through. Okay, so you run through your study design, you decide this is what we're going to do, this is going to be a great way to answer a question. Who are you going to call? So this is probably one of the things that opened my eyes the most when I started working on these things, is that you have these teams of 50 to 100 people. You have to get 5, 10, 12 boats out there. You have, a catch boat where a fisherman who has worked with seine nets 360 meters long, 7 meters deep, he has to understand tides, he has to understand weather, how the animals are going to behave so that you can catch these animals responsibly. You have animal handlers from local institutions, SeaWorld, from NOAA, from the foundation, who have been around these animals for a long time and know the proper way to care for them and handle them. You have people driving boats that have to know about dolphin behavior and keeping everybody safe. Of course, the veterinarians, sample processors. We basically set up one of the boats as a floating molecular biology laboratory. So you have to have people that are comfortable working in those conditions safely and manipulating those samples in real time. On and on, field biologists, data recorders, equipment managers, you can go right down the list. All of these folks are just really so incredible when you sit down and talk to them and hear their stories about what they've done in their careers.
What we're looking to get is small numbers, 1, 2, or 3 animals in workable waters, which is anything less than 5 feet in depth.
We've got a blue tag with 2 or 3 animals coming your way, Eric.
Copy that, yeah, I just picked them up at my 12 o'clock. I see them.
If we find such a group, we'll call in the catch boat with a 400-yard long net on the back of his boat.
Dead out, dead out, dead out.
And we'll set a compass, basically just a big circle around those dolphins.
Yeah, I got about 3.5 on my sounder.
There's 123 in the net. We have 2 in the net.
2 in the net. 2 in the net.
We've got about 15 to 20 experienced animal handlers who will then put in the water and space them evenly around the compass and induce the animals to hit, ideally in a location that we choose.
Go forward. Forward.
The dolphin hits the net. Typically when they encounter that net, they'll spin on their long axis, which wraps them up in the net, which is actually a good thing for us because that slows them down. We'll let the net do its work, and then we get the handlers in there to support the animal and get them to the hands of the veterinarians and the trained staff who can then do the actual science of what we're doing out here.
Each dolphin receives a thorough examination, not unlike what was done back in San Diego. The major difference is that here, the goal is to discover any chronic health issues linked to oil exposure.
You guys okay? Can we do capnograph? Yeah. Yeah? Okay, so you guys can go ahead and come in and do that.
And because dolphins are mammals and breathe through their blowhole, monitoring their respiration will help the veterinarians decide whether their lungs are healthy or diseased.
48.
Blood samples are drawn from tail fins to screen red and white blood cells and to check for any abnormalities with their immune system.
Very cool, number one, right? It's amazing to see in person. Number two, I just want to make sure, again, to emphasize that the decision to actually catch these animals is excruciating, especially when you're out on a boat in 95 degrees in the humidity in Barataria Bay, waiting for something to happen. But in this case, Eric Zollman, the gentleman that was talking, is the person who's in charge of basically making that decision. And so that's him standing in the background there. And you know, he's thinking about so much at the same time, right? Number one, we can only catch so many animals safely at once. Number two, we want to make sure that they're old enough to handle the stress. Number three, we want to make sure that they're not too far along in a pregnancy to risk the pregnancy. We need to make sure that the water's shallow enough for the team to do their work safely, that the bottom of the seafloor is going to be safe. Weather, you know, just a ton of different parameters that go into making sure that all of this is done with the utmost care for both our research personnel and the animals. Okay. So, we did it. A bunch of apes in water got an amazing aquatic animal into the veterinarian's hands. So what do? It's actually very much like if you and I go to the doctor for a physical. Maybe more like a professional athlete. But you get lengths and weights on the animals. We take a picture of their dorsal fin to help with those photo ID cataloging, and just in case an animal shows up stranded and dead on a shoreline, we can help identify what that animal, who that animal might be. We look at their teeth. We collect information on perhaps breath and COT levels. We look at heart rates and different cardiac assessments. We draw blood and do an amazing amount of laboratory analyses on these blood samples. And down on the right, that's just the sample processing team working. Since this effort, we've actually gotten another boat that's actually enclosed to make the sampling a little bit more streamlined. So this is a more comprehensive list of the types of things we can do. We can look at contaminant load. So if you're looking at a PAH exposure, you're looking at a PCB exposure, we can start looking at that. Just to give you an example, Randy Wells over in Sarasota has been doing his health assessments for over 20 years now, which is another incredible fact. And as long as those animals are unstressed and stable, which a lot of them are pretty used to getting captured at this point, last year I think he said he could have done up to 43 research projects on one of these animals. Now that's not, you know, every scientist lining up waiting to get their hands on the animal. This is a combination of people doing things at the same time, taking samples and then taking splits of those samples and all working from the same samples. And it's a combination of people doing things right there in the field, at the lab, in the evenings in the field, or sending it off to external laboratories to do their analyses. Okay, so once you have all of that data, what can you do? So Lori did a great job of summarizing some of the effects that we saw for the Deepwater Horizon injury assessment, and I'm just going to kind of make that link a little bit more clearly about how health assessments directly informed some of those findings. So when folks were out on the water, they kind of anecdotally said, ooh, these animals look skinny, they don't look very healthy. But it's not until you actually get them in hand, right, and you can start doing mass to length ratios and comparing animals in Sarasota Bay, which are in blue, which in Florida they didn't get any of this, in Sarasota they didn't get any deepwater oil, and compare them to animals in Barataria Bay. These are the red dots, and so for the males you can see really had poor body condition, and the females kind of were straddling that line of what's considered a healthy body condition. As much as we try to not stress out the animals, there's inevitably going to be some stress response, right? When you're setting a net around these animals and you're grabbing them. And this should, in a healthy animal, induce some sort of cortisol release into the bloodstream, kind of your fight or flight response, right? And so in Sarasota, you can see that when we collected blood and we sent it off to the lab, you can see these sort of elevated cortisol levels. But in Barataria Bay, these animals didn't induce that stress response. They had very, very low levels of cortisol and aldosterone. So this is, again, something you're not going to be able to do with a photo ID survey. We talked about the ultrasound earlier, and so this is something that without having a vet with a lot of expertise, with their hands on an animal, you're never going to know that Barataria Bay dolphins were 5 times more likely to have moderate to severe lung disease. You're looking at the pleural lining of the lung, and you can start to see, if you're one of these amazing vets, that the tissues lining the lungs are starting to get inflamed, you're starting to see edema, and this is going to impact how the animal can deal with additional stressors in addition to the lung disease. And then using the ultrasound, just like with human pregnancies, we can start to look at fetal and neonatal loss. Normally the data that are associated with looking at reproductive failures is something that you're doing with reproductive follow-ups and photo ID surveys. But now, if you have the ability to look at some of these animals, number one, you can identify early-term pregnancies that you might have missed. Number two, if you look at the diameter of the skull, you can actually predict the due date. And so you can help your photo ID teams and your follow-up teams by saying, okay, you probably need to find this animal around this time to identify whether this pregnancy actually came to term. Okay, so since the Deepwater Horizon NRDA, we are following up on those reproductive effects of oil exposure. I think Lori mentioned this, but it's worth repeating. In Sarasota, they typically have an 80% reproductive success rate. In Barataria Bay, post-deepwater, was 20%. So we're trying to figure out, are those effects still happening? It seems like they're starting to come back up, but that's the work that we're doing right now for a GOMRI 4 project. We're also working closely with NOAA and the state of Louisiana to pair health assessments with satellite telemetry to track dolphin movements in Barataria Bay, to start to inform decision makings about potential Mississippi River diversions. And then what I'm going to try to fly through here at the end is our exciting new project, the Consortium for Advanced Research on Marine Mammal Health Assessment, CARMA. So CARMA is a consortium of something like 10 institutions, all research scientists, and we have funding for 2 years to basically improve our veterinary and laboratory techniques to assess dolphin health. We are also going to look at the indirect effects of how Deepwater Horizon might have affected prey and therefore had effects on dolphins. We're really interested in looking at the health status of dolphins from stocks other than Barataria Bay and Mississippi Sound, which are the 2 that we mostly looked at for deepwater. So we're going to get out there and look at the northern coastal stock. And then we're going to pull all of that information together and anything that we can find recently from Gomery and other sources to improve our models of population recovery trajectories. So we're focusing on 3 different thematic projects: cardiac assessments, immune assessments, and dietary and trophic analyses. So since the NRDA, some of the veterinarians have noticed that anecdotally they're hearing what seems to be heart murmurs in the dolphins. And this is particularly interesting because a lot of the NRDA work showed that birds and fish, when exposed to Deepwater Horizon oil in labs, had some pretty serious cardiac abnormalities. So we're going to follow that up. Our colleague at UConn, Sylvain de Guise, has found that there are some immune dysfunctions going on with different pathways in the dolphins that we captured. And so there's not a lot of assays and reagents that are specific for dolphins at this kind of cutting edge of immunology. So he's going to work on developing those assays and apply them to samples that we collect. And then Ruth Carmichael at the Dolphin Sea Island Laboratory and Maggie Broadwater at NOAA NCOS are going to look at stable isotopes and fatty acids respectively to try to get at this question of how diet and how body condition might affect health a little bit more closely. And so, of course, we're going to use these, we're going to use field studies to help answer some of these questions. So we'll be doing some trawls associated with our health assessments for the dietary sampling to look at prey samples and collect dolphin samples. And then we're going to do 3 different health assessments. We're going to continue working with Randy down in Sarasota Bay, one to make sure that the techniques that we're developing can be deployed in a little bit nicer of a setting in Sarasota. We're going to, and collect data on a reference population. We're going to continue our studies in Barataria Bay, which is going to be led by Terry Rawls at NOAA. And then we're going to look at the northern coastal stock, like I said, which is going to be led by Brian Balmer at the foundation. So just real quickly, you can see that this coastal stock actually these animals can move all the way from the bend in Florida all the way to the mouth of the Mississippi River. And so one thing that we're going to have to pair these health assessments with, since we're going to be catching at Dolphin Island, the red circle there, we don't necessarily know whether the animal we catch is going to be from the coastal stock, which is our target, or whether it'll be from the Mobile Bay BSE stock, Bayside Estuary, or whether it'll be from the Mississippi Sound stock. So we're going to have to follow up our health assessments with satellite tagging and with remote biopsies combined with genetics to really dial in, okay, what family does this dolphin belong to, basically? And is it one of the animals that we're trying to identify with this northern? So real quickly, we're going to use these data, combine it with literature searches, synthesize it all into one place. We're going to use a technique called expert elicitation to kind of translate some of these health parameters into what we think that means for survival and reproduction in these animals. And then we'll use those to improve the models that we came up with for the NRDA. And so this is a simplified version of what Lori showed earlier, where the reduced survival factor and the reduced reproduction factor kind of give you what is the effect of Deepwater Horizon on this stock. Here, this is Barataria Bay, control and affected. But now with these additional parameters, this additional time to pull everything together, we think we can refine these things. And just to emphasize that we are not just interested in BSE stocks, Barataria Bay, and Mississippi Sound, but one of the things that we want to do with Karma especially is start to get a better handle on what is going on with these oceanic stocks, these shelf stocks, and these coastal stocks. Thank you.